Thank you, Sarah, for the introduction. So my talk, I made it thinking that it would be like a giant introduction for Marina's talk. Okay, so feel free to ask questions at the end if you need more details. What I'm going to present is basically what is an epithelial tissue, why we should care about what is an epithelial tissue, not just because we study that in the lab, but why is it important to understand how it works, and also how dynamic it is, okay, and why this dynamic is difficult to tackle, and why having a project at the interface with mathematics will help understand better these dynamics, at least we hope. So from this, I'm going to give you a bit of background on epithelial tissues. So here on this slide, you can see a single cell as part of an epithelium. So that cell in the middle here is attached to neighbors. So you have a cell on the right and a cell on the left. And these cells are attached to each other, and they are polarized along what we call an apicobasal polarity. So the apical pole will be here on top and the basal pole at the bottom here. And if we zoom in at the interface between two epithelial cells, you have a lot of structures that are represented on that scheme at the top. That basically bind the cells together. So it doesn't really matter the type of protein we are talking about. Let's think that you have some kind of glue basically that is in between the cells that is formed by these bridges of proteins that are part of the membranes of the two neighboring cells. And by attaching to each other, they bind these two cells together. So that's one of the main characteristics of epithelials. That you have these cells attached to each other, being polarized. On one side, you have this apical pole toward a free space or a lumen, and then this basal pole attached to an extracellular matrix. So nearly all tissues are epithelial at some point. Okay, in early development, when multicellular organisms are forming, all tissues are initially epitheliums, and they will go through changes during the life of the embryo while the different organs are forming. So understanding epithelial dynamics is actually relevant to all sorts of tissue, even if in the adult, some of these tissues are no longer epithelial, okay? So that was a zoom on one cell. If we look at cells being together, you can form a tissue, and the shape of this tissue will be influenced by different forces that you have in the tissue. You have the interface with the basal, with the matrix on the basal side. So the more you interact with this matrix, the more you will flatten on the substrate. And you have also forces at the apical pole, here represented by this actin cable that is contractile. And the more you contract it, the more narrow the cell will be. So depending on the balance of these forces, this is a simplification, obviously, you can vary the width of the cell, the height of the cell, and then the shape of the epithelium. So here are some examples of different types of epitheliums you can get depending on how these forces are playing out in the tissue, from a simple squamous epithelium with flat cells to more complicated one like stratified columnar here on the bottom right. And if I go back to that slide I had as background for my title, here you can see an example of 3 different types of epithelium. So on the left side here, this is a cross-section in an amphibian embryo. You have the skin, so this is the external layer here with very flat cells. So this is like a squamous epithelium. You have a layer of matrix in between here that we don't really see with this magnification. Then underneath you have these cuboidal cells. You have 2 layers of cells. One is more cuboidal, the other is more flat. And then underneath that you have, on the bottom right corner of this image, you have elongated cells that are pseudostratified. So all these cells are very elongated from their apical pole to their basal pole. And they are organized as shown here on the side with nuclei at different positions. Okay. So these structures are actually quite transient in development because epitheliums have to be disassembled at some point so that cells can move around and form the different organs. So there's a lot of plasticity, which is represented here on that scheme. You have that initial epithelial cell that I was showing you a couple of slides ago, and this cell can remodel its organization to adopt a different fate that is called mesenchymal, that you have here on the right. And the mesenchymal cell, instead of having a strict polarity with apical and basal pole, has a more flexible polarity that is called front and rear polarity that will adapt to the environment and allow these cells to pick a direction and migrate. So to go from this apicobasal polarity to this front-rear polarity, you need to change a lot of things. Some of these changes are listed here. These core epithelial mesenchymal transition changes. So you need to change the cytoskeleton, you need to change the adhesion between cells, you need to change the adhesion with the matrix, and so on and so forth. And what's important is that this is reversible. You see all these arrows are double-sided, so you can go from epithelial to mesenchymal and go back to epithelial features. And you can also have these intermediate states where cells lose some of their epithelial characteristic, but keep some of them, okay? So this is what we observe during embryo development, but also what you observe during wound healing. For example, when you have a cut in the skin, the cells will go from this epithelial state here to a more mesenchymal state here, where you have a breach in the epithelium, and then within this gap, the cells at the edge will form this protrusion and adopt a pseudo-mesenchymal phenotype so that they can migrate and close the wound. And we can also observe some of these changes during cancer metastasis. The typical framework you would have in this case is the following one, where you have a primary tumor in an epithelial tissue. And then within this tumor, you have some cells that are losing epithelial characteristics. Some of them will make a complete conversion to mesenchymal state, will start migrating. Then they can use the environment around them to travel across the body, settle in a new location. For example, here, after traveling through the blood vessel, they will settle in the lung, and then some of these cells will revert to a more epithelial state. And so you have this, over time, this plasticity that will drive cancer metastasis. But this is very difficult to study in situ, obviously, because of doing experiment in cancer patients is going to be extremely challenging. So there are alternative models to work on that. For example, like during embryo development. So here is an example of what happens during the development of a chicken embryo. So at the bottom, you have a Holman view of the early embryo. At the beginning, it's just a flat disc. If you make a cross section, you can see that this disc is a form of an epithelium. And then in the dorsal region of this epithelium, some cells will convert into a mesenchyme. Those are these small blue cells that we call the mesoderm, and these cells will migrate, and then later on they will reaggregate and form a new epithelium that is displayed here in yellow, that is called the somite. So you can recapitulate this epithelial mesenchymal plasticity that I was showing you during cancer metastasis at early stages of chicken development. So this is just one example that can be used for experiment. Another famous example for this is the neural tube and neural crest cells. So this is what we use mostly in our lab. Here you have a diagram showing the basic organization of an early chicken embryo with the head on top and the trunk at the bottom. This big circle here is the eye, and the dorsal part is on the left side of this slide. And what you can see if you make a section of this embryo here at the level of the red line, you have a structure here with the skin on top, the neural tube, which will form the central nervous system, and then the precursor of the muscles that are called the somite on each side. And what's interesting is that in the dorsal part of this epithelium, the gray area here, some of these cells are undergoing this conversion from epithelial to mesenchymal to allow these cells to migrate and then colonize different regions of the embryo, very much like cancer metastasis cells does. And then they will go on and differentiate and give rise to a lot of derivatives in the body. Okay, so this is a nice experimental model, and it's been used by a lot of different labs to study this plasticity between epithelial and mesenchymal state. So we now have a lot of information about what occurs in this gray area when the cells are converting from epithelial to mesenchymal. So I'm not going to go through this type of slide. It's just to show you the kind of network that we've been building by experiments, trying to knock down some genes that are expressed in these cells, overexpressing them, comparing the effect on the epithelium when we block them, when we activate them, and we end up with this type of complex network of genes that are interacting with each other with different types of protein involved. So you have what we call external signals that are proteins that are in the medium, in the environment of the cell, that are secreted, like this BMP or Wnt, for example, that are here on the left side of the slide, that will control the expression of genes that are within the cells that are called transcription factors, like this snail or SOX, that are represented here in the middle of the slide. And then these genes will regulate expression of downstream effectors, such as cyclins to control proliferation, cadherins to control cell-cell adhesion, extracellular matrix component and integrins to control the adhesion to the substrate, and component of the cytoskeleton to control cell shape and dynamic of the membrane. So it looks like we understand a lot. So we know how it's triggered, what controls the expression of this gene, and how it's implemented in the cell. We even go then further, because the cells, once they have converted to a mesenchymal phenotype, they will migrate. And there you have a whole different ballgame of interaction between proteins to control the cytoskeleton of these cells and how they will move, how they will respond to different signals in their environment and so on. So by looking at this, we could think that we understand how the cells are converted from epithelial to mesenchymal. We know how the gene regulatory network is implemented to control the downstream effectors, and it looks like we know everything we need to know. But actually, it's not the case, and there are a lot of problems with this, and that's where the complexity is the greatest. Not all cells are actually expressing all these genes I mentioned. So we identify all this network by experimentation, by gain and loss of function, but it's not always what is in place. So you may have cells that are only activating part of the network. You also don't have a strict order of events. So this network that I showed, you had green arrows for positive action, red arrows for negative action. So you have a lot of complex different feedback loops. And so if you think of a cell in an epithelium that is attached to its neighbor, the basic way that you would want this cell to detach, if you were doing it as an engineer trying to plan something strict, you would say, okay, let's first detach that cell from its neighbors. Then change the polarity of that cell so that it can orient itself in the environment, and then promote some motility so that it can move around. So that would be a logical way to do it. But the cells are not following logical steps like that. So you can have cells that follow this path that I just mentioned, but some of the cells just activate migratory phenotype first without caring about whether they are detached from their neighbors. So you have all sorts of scenarios to go from an epithelial tissue to a more mesenchymal tissue, and that is very tricky to address. Then, as I mentioned, there is plasticity. So you can go from an epithelial to a mesenchymal state, but you can also stop somewhere in the middle. So you have cells that have lost, for example, their cell-cell adhesion, but that are not motile. You have cells that display very active membrane dynamics, but are still attached. And so you have all sorts of in-between phenotypes. So to make it short, there is heterogeneity, and that is the main issue that we hope the interface with mathematics will help us understand better. So the associated question with this heterogeneity that we can see is what does it mean in our system to have a complete or a partial conversion to a mesenchymal phenotype? That is not so clear. And then if we have a collection of cells with different phenotypes, do they cooperate? Is there synergy? So is this important to know if one cell is able to do something versus what its neighbors are not able to do, and whether they will cooperate and go on and migrate together, for example. And then if we know that we have different ways of undergoing EMT, and we end up with a collection of cells with different phenotypes along that spectrum from purely epithelial to purely mesenchymal, does that matter for what they do later on? So does it influence their ability to migrate or to colonize specific tissues? Okay. And this heterogeneity that we've seen in embryos is also true, and even worse in tumors. So this is actually critical to understand the importance of this heterogeneity if we want to address the problem of how cells will spread from a primary tumor, for example, and settle to other organs. So how can we challenge this epithelial stability, and what kind of experimental approaches we can use to answer some of these questions related to heterogeneity? So as I mentioned in that slide with the network of genes, there are three main levels at which we can address this problem. One is the extracellular signal that the cells are receiving. One is the transcriptional level, so the gene regulatory network, how gene expression is activated or repressed in the cells. And then the downstream effectors, so the actual proteins that are regulated that will modulate adhesion, polarity, proliferation, interface with the matrix, and so on. So in the lab, we focused more on second and third option. And the final readout is when we challenge any of this player in the network, is can we trigger extrusion of cells from the epithelium? So do we prevent extrusion in a population that should have come out and start migrating, or can we trigger extrusion in an epithelium that was stable and was not producing mesenchymal cell at the time? So how can we do that? So one of the models we use is the chicken neural tube. So on the left, you have a drawing of a chicken embryo at an early stage. And the neural tube, as the name indicates, is a tube. So there is a lumen in the middle, and we can inject into that lumen a solution containing expression vectors for various genes or tools to perform loss of function for the genes of interest. So this is represented in a bright green color here in the middle. So with a micropipette, you inject a solution containing the genes you want to activate or the tools to modulate their expression with a fluorescent tracer so that you can monitor which cells have received your gene of interest. And here you have a picture on the right of a chicken embryo the day after this injection. So what you do is you micro-inject in the lumen, and then you put small electric pulses to destabilize the cell membrane so that the cells will take up whatever you injected in that lumen. Then you can do some histology and analyze the architecture of the tissue. So this neuroepithelium is what we call a pseudostratified epithelium with very elongated cells, as is represented here. And the nuclei are moving along these very elongated cells between the apical pole and the basal pole. It's a very dense tissue. So here are some examples if we try to modulate or to challenge epithelial stability by playing at the transcriptional level. So here we express different combination of genes that are upstream of these final regulators of adhesion and polarity. So on the top row, you have the effect of expressing a gene that is called ETS1, which is a transcription factor. The first image here, the DAPI picture, shows you all the nuclei you have in that tissue. So you can see in the neural tube in the middle here, it's a very densely packed tissue with hundreds of cells on top of each other. Okay, then the second image, the GFP, is the area where the gene was electroporated. So all these white cells have received ETS1, and the other one on the left side have not. So it's the control, the internal control. Then we have staining for laminin, which is a molecule of the extracellular matrix. And you can see where those two arrows are pointing that you have holes in this matrix. So usually you have a thin line all along the tissue. And here in the area where the gene is overexpressed, you have holes that are created. Okay, and you can see some of these green cells here popping out from the tissue and crossing the area where that line should be. But this is kind of a mild phenomenon that we don't have like hundreds of cells leaving out the epithelium, okay? If you co-express that gene with another one that is called SOX9, which is what we see at the bottom of this slide, you have, again, holes in the extracellular matrix. You see where you have those three red arrows. It's almost completely gone. And then at the level of these holes, you have green cells that are popping out into the environment here. If we look again at these two experimental conditions, but with other markers. So if we look at an integrin, so this is the third column here, you can see in the co-expression of these two genes that you have a massive expression of one of the integrins. So it's a receptor for the molecule I was showing you before. So this is the laminin, which is the extracellular matrix receptor at the basal side of the cell. And this is the integrin, which binds to it. Okay, so it's essential for the cells to attach to this matrix. You can see on the left side, on the control side, you have just a line, a white line of this integrin activated at the basal side of the cell where they bind to the matrix. But when you overexpress these two ETS1 and SOX9 transcription factor, you get activation of this integrin everywhere. And the epithelium is completely messed up, the cells are round. And then if you look at a molecule that binds the cells to each other, in this case, one that is called N-caderin, and again, you can focus only on the third column here in the middle. If we overexpress ETS1, we don't see any massive downregulation of this protein, but when you co-express it with SOX9, then you have these black areas full of green cells, and if you look at the green cells on this image, you can see that you don't detect any signal in them, so that means they have lost this cell-cell adhesion molecule. So based on this diagram I showed you at the beginning, where you have a spectrum between epithelial and fully mesenchymal, the phenotype we have with ETS1 is like a partial EMT. So the cells, they still have N-cadherin, they are still attached to each other, they are kind of clustering, but they are not really able to leave the tissue. They make holes in the matrix, but that's it. But if you look at the ETS1 plus SOX9, then you have a more massive phenotype. You have a downregulation of cell-cell adhesion, you have activation of the receptor for the matrix, you have holes in the matrix, and you have cells that are successfully leaving the tissue and start migrating like these few green cells I'm showing you here. Okay, so that's just a zoom to show that these green cells don't have any cadherin. Okay, so here we have, so by making a lot of different comparisons and testing different cocktails of genes, we ended up selecting these two as example of partial EMT phenotype and complete EMT phenotype. And from there, we made a screen comparing with two controls, one with just the fluorescent reporter alone that we use as negative control, and one with SOX9 alone, which has no effect on the epithelial structure. From this, we purified our tissue of interest and we ran a transcriptome. So we had the full list of genes that are up and down regulated in these cells. And what we found is that we have different signatures for the control cells, the pseudo-mesenchymal cells, and the fully mesenchymal cells. In particular, we found some different signatures in a family of proteins that is called matrix metalloproteinases. I'm not going to give a lot of details about this. I just want to show one example. We are studying a lot now the function of this protein because we think they are key players in this in modulating the switch from purely epithelial to mesenchymal. And we started releasing some papers and preprints this year. So if some of you are interested specifically in that, you can have a look. I just want to show an example. So if we pick one of these metalloproteinases that we found to be important, which is called MMP14, and we look at transversal section of the chicken neuroepithelium, and then we stain. So again, the green cells have received that gene, the other ones don't have it. And we look at position of cells that are dividing. If you see on the left side of each image, you have those purple dots that are aligned here on one side of the tissue. But then if you look in the green area, you have some of the purple dots that are on the opposite side, like where you have those white arrows. You can see some of these purple dots that are on the basal side of the tissue. And this is indicating that when we overexpress this MMP14, we have a problem of polarity. So usually you have a nice apicobasal polarity and all the dividing cells come to this apical pole. But when you start messing up with this enzyme, you challenge the apicobasal polarity and you get what we call ectopic mitosis in the basal side. So this is to show that we can test then experimentally all the candidates we find in this transcriptome that specifically associate with a partial mesenchymal phenotype or complete mesenchymal phenotype, and go back and express them or knock them down in the epithelium and check the consequences. So these ectopic mitosis are indicative of an apicobasal polarity problem. And you can then compare this type of phenotype with what you get if you really challenge apicobasal polarity by targeting a key player in this process. In this case, I'm going to show you an example with PAR3, which is an apical protein in our tissue. And usually it's found only along the apical pole. And here we massively overexpressed it to completely confuse the cells and make them believe that the apical pole is basically everywhere. Okay. And if we look at the consequences, they are dramatic. It's much more dramatic than overexpressing that protein I showed you the slide before. So here you have massive reorganization of the epithelium with this big mass of cells protruding into the lumen. The matrix doesn't seem to be affected. If you look at cell-cell adhesion like N-cadherin, you have partial loss of N-cadherin here. You see a big interruption of this staining and accumulation on the basal side. Okay, so it's like the cells are completely flipped. They lose the cadherin on the apical side. They start accumulating on the other side. So they are not able to maintain their normal polarity anymore. If you look at mitosis, so here the red dots are cells that are dividing. Instead of having one line of dividing cell on the apical side here, you start having cells all over the place. If we zoom in, you go from this nice apical alignment of mitosis to mitosis being completely distributed randomly in the tissue with lots of them in the basal pole here. So by comparing this loss of apical polarity with this master regulator polarity with the other gene that we had before, we can start placing them and building new networks of interaction between the protein of interest. Okay. So this is just to show what we can do globally. But what I told you is that we have heterogeneity. So this is the main issue. So what does it mean? So we could have two cells that have different levels of mesenchymal phenotype. For example, you could imagine one cell has lost here, cell A has lost cell-cell adhesion, and the other cell next to it has lost apicobasal polarity. So what happens next? Is that sufficient to create extrusion? Is that sufficient for the cell to exit the tissue? How can we reproduce that experimentally, and how can we analyze this type of stuff? The other type of heterogeneity is over time. You can imagine that one cell is going full mesenchymal transformation, but follows this step in a specific order, first losing adhesion, then polarity, then degrade the matrix. While other cells will do it in a different order. They will lose polarity first, they will make a hole in the matrix, and only after that they will detach from their neighbors. So how can we control this different type of situation? So one way is to do this electroporation that I showed you before, but to do consecutive electroporation with different doses. So here, if we make a test, for example, with different markers, green and red, just as a proof of principles, and we check the tissue, so we can inject a fluorescent tracer green, zap the cells, they incorporate it, then do the same with a red one. And if we play with the different concentration and the voltage, we can generate this heterogeneous population where we would have non-transfected cells that are not fluorescent, some green cells, some red cells, and some yellow to orange cells that have received the two genes of interest. So with this type of technique, you could generate mosaicism in the tissue. And then you could follow the cells over time and check what happened with the green cells that have received gene X, what happened with the red cells that have received gene Y, and then with the yellow to orange cells that have both of them in different ratios, and compare their behavior and their position in the tissue and so on. Now, the problem is that we have thousands of different scenarios to test, and we have lots of genes that are involved. Okay, so also you have technical difficulties in terms of calibrating the experiment to make sure you always have the same number of cells, the same distribution, the same density, and so on. So this, if I was to try to target that solely by biological techniques, it would be just completely stupid, because we would have so many things to test, it would be just crazy. So, and on top of that, you have a lot of interplay between biological parameters. So none of the experiments you can do in biology is going to be 100% clean, because I showed you these networks with lots of genes, but the thing is that you have to think that one gene is participating to this process, part of this network, but it may have also other functions that are not related to this, and you won't be able to specifically restrict that gene to your problem of interest experimentally. For example, a lot of these genes that are involved in mesenchymal transition, they are also involved in acquiring specific identities and specific functions later on. So that is tricky to address. So that's where the modeling comes in, because we hope that by modeling our tissue, we will get fine-tuning at single cell level, and that we will use that then the readout of the tissue shape to make assumptions about what is the best strategy to try to tackle biologically. So to finish very quickly, this is the expected framework we are aiming for, with on the left side of this slide the biological strategy, where we can make a global assessment of the gene functions, generate mosaic population, or do sequential activation of genes or loss of function over time by doing this technique of electroporation. Then analyze the tissue, perform different staining for the key factors in our tissue of interest, then analyze this and define what are the best scenarios. But to avoid having thousands of scenarios to test here, we hope that the computational approach will help us refine this. Okay, so what do we need to get this side of the framework? First, we need a model of the epithelial growth that matches our experimental system. So this is the very dense neuroepithelium I showed you before. Here we see it growing over 2 days. And these cells within this tissue are very thin and elongated, and all the nuclei are packed on top of each other. So the challenge was to generate a model where we can reproduce this packed organization of very elongated cells and let them grow over time and make sure the tissue has the right size and shape. And then from there, hopefully in the second phase, implement control of parameters of adhesion, polarity, and proliferation at the single cell level to try to model the different scenarios to go from epithelial to mesenchymal. So I'm going to hand it to Marina here. I just want to mention the people from the lab that have been contributing to this project, Fernando, Evangeline, and Cyril, and the collaboration with the mathematician that started with Pierre Degon and Marina, initially at Imperial College, and that has been followed up now with Sarah and Stefan. And I guess Marina will mention a bit of that in our presentation. Thank you, and I leave the talk to Marina.
Thank you.
So I can share my screen. How do I do that? Yeah.
Hello. Can you see my screen?
Yeah, I can see the screen. Is it still not full screen? Yes, perfect. It's full screen now. Okay, thank you.
Okay. Yeah, first, thanks for organizing this workshop and for the invitation. So I will talk about the work I did during the PhD with Pierre and Sarah. And yeah, in collaboration with Eric's team in Toulouse. And I will mainly talk about the how we build the model of the epithelial tissue. So the main aim was to build a mechanical model to take into account the contact forces between cells in a crowded environment. So as Eric mentioned, these epithelial tissues are very packed, and the dynamics of individual cells can have an impact in the whole structure of the whole tissue. So this is the main question: what is the role of this crowding and forces to the global shape and stability of the healthy tissue that we want to address this question. So to build the model and to take into account the effect of the contact forces, we need to consider cell volume and individual cell and describe the cells individually. So we use an individual-based model, and I divided the models into complex particle shapes and simple particle shapes. So in the literature, we find cellular pods model, subcellular element model, Voronoi or vertex models. The first two, they are based on complex cell shapes, and the last two, they have crowded already in some sense in the model. So it is not appropriate to study our problem, because we would like crowding to be an emergent behavior. So then what we consider is simply spheres to model the cells. And in this case, there are works in the literature where spheres or points have been used to describe different scenarios in biology, birds, cell migration and proliferation, or tumor growth. And an important reason for choosing simple cell shapes is that they allow more easily to derive macroscopic models and have a connection between the different scales, which is important in these complex systems, to keep consistency between scales. And so this is our main philosophy for the modeling choices. And we try to keep the model as simple as possible. So we focus in three main rules. rather than trying to model every mechanical aspect of cells, what we do is we consider behavioral rules. So the first is that particles cannot overlap, and this is important to address the effect of crowding, and also to prescribe a minimum size for the cells. Then particles seek a minimum of a given confining potential at all times. So it's like we assume that the tissue is always, or the cells are always trying to release strength and reaching a minimum of some energy. But of course, because the potential W will be evolving over time, then a new equilibrium, a new minimum of energy will have to be found locally. Okay, and this the evolution of this confining potential is due to the evolution of particle characteristics such as size, for example. Then to implement this model, we will use a time-stepping algorithm, and this is summarized in 3 steps that we do within each time step. So we first actualize the positions that leads to motion. Then we actualize particle characteristics, and finally we solve the overlapping that we might have done during the first step. And this is a critical point because it's the most computationally heavy, and it involves the way we do it. We solve a minimization problem with constraints, because we want to ensure that the particles have do satisfy hard constraints. Okay, so I will first describe the tissue at equilibrium. So I will describe the potential and the constraints and how we tackle the minimization problem. Then I will describe how the dynamics is implemented, driven by noise, cell division, and changes in cell characteristics. And finally, I will show the experiments in the healthy tissue. Okay, so I have here the chick embryo. As Eric mentioned, this is the biological model that they use in the lab, and a cross section of the neuroepithelium, which is represented here. Here is in the basal membrane and the apical network in the middle here. What we see is that the nuclei are very packed, and also they will move up and down inside the cell in order to perform division. So the nuclei, as Eric mentioned, they always divide apically, and for that they travel inside the cell to the apical site before division. So I will start by describing the energy that we chose for the cell, for each cell, and this energy is related to the springs that. So the model that we consider for a cell is given by a nucleus that is the most rigid part of the cell, and a basal point and the apical point that are connected to the nucleus through springs. So the nucleus has an inner part that is a hard radius, a hard core, and the outer part that is a soft core, and this gives flexibility to the system. and models the formation of nuclei. And then the springs are between the nucleus and the apical and the basal. So we will assume that the basal point travels in 1D in the basal membrane, and the apical point can move freely in 2D. I will then show the rules for the apical point, but so far these two springs, they are the energy, we consider linear springs, so the energy is given by this, where at the IA and at the B are the rest length. So if the cell is at equilibrium, then this W is 0. And it means that the position of the apical and basal point are exactly at the distance at the A and at the B from the center. We also impose an alignment force and between these 3 points, and this ensures that the good configuration of the cell. Okay, so then two cells interact through the apical side. So there is a spring, and this is modeling the connection between cells at apical side. This spring, again, we consider a linear spring with rest length A0. And we consider also the energy of the soft nucleus that is given here. Okay. So the constraints are the non-overlapping constraints between the hard core. Phi is given by the sum of the radius minus the distance between the centers. We want this to be negative, meaning that the two centers have to be farther than the sum of the distances, the sum of the radius. Also, the basal points, we assume that they keep an order, so they cannot switch between each other. So psi is negative, and also they cannot be too far away from each other. So this is this constraint here, and this models lateral adhesion. Okay, and we collect all these things together and we obtain a minimization problem with non-overlapping constraints and the non-switching and lateral adhesion constraints. Then we want to solve this problem. So I rewrite it here just in the case of this constraint, because this is what the non-overlapping constraints are the main difficulty, since they are non-convex. Phi is a non-convex function. Then to solve this problem, we first use a Lagrangian formulation, where we write the Lagrangian as the sum of the potential with the constraints times the Lagrangian multipliers lambda. And the necessary conditions of optimality say that if x bar is a solution of the minimization problem, so if it's a minimizer, then there exists a lambda bar such that x bar lambda bar is a critical point of the Lagrangian. So it means that it satisfies the KKT conditions. And in the case of convex optimization, this is also sufficient. These conditions are also sufficient. And this is the idea of the famous, well-known Arrow-Hurwicz algorithm that uses the fact that so it searches for a steady state of a system that uses the information that the solution should be a saddle point, meaning that we should minimize in x and maximize in lambda. Lambda represents like the intensity of the repulsion forces that ensure the non-overlapping between the hard core of the nuclei. Okay, and so this system goes in the direction of minus the gradient of the Lagrangian with respect to x and plus the gradient of the Lagrangian with respect to lambda, aiming at finding the saddle point. This works well in the case of convex problems, but in the case of non-convex, it may not converge due to the existence of periodic solutions. So it may not converge to the stationary state. And to solve this problem, so we found this was a problem in our case, and what we did was to change the dynamics of the system in a way that we do not change the steady states, but we change the stability of the steady states so that they become asymptotically stable. So for that, we consider the second order formulation of the previous ODE system, and we added a damping term to damp out the oscillations. This was done in a way that we do not change the steady states, as I said. So we can see this because at equilibrium, the second equation is the same, and so what? Okay, comparing to the previous system, the only difference is the first equation. And at equilibrium, the second equation implies that phi times lambda is 0, and so this term is 0 at equilibrium, and we have just that the gradient of L equal to 0. This is what the equilibria in this system satisfy, which is the same in the previous system. So the two systems have the same steady state solutions. Now, just a few remarks. We did some study in a simplified sphere packing problem, and we realized that, we showed that the damped Arrow-Hurwicz algorithm converges in 1D, and in 2D and 3D, it performs reasonably well for a large number of particles. However, it can be improved in terms of numerical convergence, because these contact problems, they are associated to stiff ODE systems, typically due to the repulsion forces associated to the non-overlapping constraints. So these lambdas, they are actualized in some situations, they are very actualized very fast compared to the energy. And therefore, this may lead to numerical instability, which requires a small step size. One possible improvement would be to consider adaptive step size to speed up the numerical convergence, or other alternative using alternative minimization algorithms like position-based dynamics, which seems to allow a much bigger step size, 1,000 times larger, at the expense of possibly violating some constraints, which seems negligible in our problem. And this is a current work by Stefan and Sarah to study this algorithm. Okay, so we keep I collect here again the minimization problem. with all the terms. So this is our energy. Now I have here a few parameters. Alpha, these are the stiffness of the springs. Also, these rest lengths, eta, and A0. So how do we choose them? The stiffness of the springs will be chosen relatively to each other, which is reasonable because what they do is that they determine which minimizer the system will find. So in fact, the problem is not unique, does not have a unique solution. We want a solution that is local because cells are not jumping to the other side of the tissue to find the optimal solution. And also we consider then this the minimizer that favors some springs with respect to others. This is motivated by biological observation. That is, for example, the apical springs, they are much stronger than the nucleus springs. So this alpha A will be of order 10, let's say, while the nucleus springs will be of order 1, for example. And this will, in the minimization algorithm, it will favor the finding a minimum of the apical energy, the energy of the apical springs. Okay, and by considering different parameters, we can observe the influence in the minimizers that we obtain. So if the stiffness of the nucleus is very large, then the tissue is quite short. If the stiffness of apical springs is small, then the apical network is quite loose. A small alignment of forces messes up with the colors. So we see here that cells are aligned, so the apical nucleus and basal points are aligned. This is represented by the same color, each cell. While if the alignment is not strong enough, then they will not be aligned. Cells will make angles. And if the soft nucleus is hard, then the tissue becomes more stiff. Okay, so this is a qualitative choice for the parameters, and then we obtain this solution. Okay, then the rest length of the parameters of the springs, they will be chosen dynamically in time. So we assume that the springs are adapting to the local environment, and this was a critical observation to that allowed the model to finally reproduce the dynamics of the tissue. So this was important, and it also gives some viscoelastic effect. So the springs are always converging. The rest length of the springs is always converging to the actual size or actual distance between apical and nucleus and basal point and nucleus. And this means that the springs are always relaxing over time. So this eta bar is the actual distance between apical nucleus and basal point and nucleus. And this is what we assume for the S phase and G1 phase. And this gives a passive movement of nuclei. Then during active G2 and mitosis, nuclei will try to approach the apical side. Therefore, we set up the apical rest length nucleus apical to 0, and the nucleus and the nucleus basal to the size of the cell, so that the nucleus wants to reach the apical side. Also, the apical spring is we consider this evolution for the rest length, where the rest length will converge to 0 exponentially fast. In the case, and we will consider the case of passive springs, where the rest length is always 0, or contractile springs, where the rest length converges to 0. And this is because we do not know how to describe this. So we explore these two options. And finally, we add the Gaussian white noise to the nuclei. And this seems important to give some flexibility to the model. So, and to find some space, yeah. So the cell stage, as I said, during S and G1, the nuclei move passively, while during active G2 and mitosis, they move actively towards the apical side. So this is represented here. Also during mitosis, we increase the radius of the hard core so that cells become more rigid. And finally, after a determined amount of time, the cell divides, and this is done by substituting one cell by two cells next to each other in this way. The duration of the stage is fixed, so the cell will divide even if the nucleus has not reached the apical site. Okay, so the algorithm, the time-stepping algorithm, consists in division of cells that reach the end of the cell cycle. Then update the rigidity of mitotic cells, update the springs, rest length according to cell stage and environment, adding Gaussian white noise, and readjusting positions using the minimization algorithm. We consider this initial condition that we generate using two rigid walls, but then when we remove the walls, the tissue still keeps a nice structure. So this is what we obtain after 2 hours of simulation, real time, yeah. Biological time, I mean, yeah. Where here is the basal points, and on top is the apical points, and in the middle the nuclei. Okay. I see that I do not have much time, so I will try to quickly give an idea on the study that we did using the model. So the first step was done by Eric's team, where they obtained data related to the shape of the tissue over time. In particular, they obtained the apicobasal length, the number of layers, and the straightness of the apical domain and position of mitosis. So this is, we can see here that the basal, here is the basal and here is the apical side. And we can see that the tissue grows along the apicobasal direction, and also nuclei seem to get more packed near the apical side and lose density near the basal side. Then the question is, what is the individual contribution of active nuclear movements prior to division, and what is the contribution of the increase in cell number to tissue shape and nuclei distribution? So we want to relate these two phenomena with these indicators of cell shape, of tissue shape and nuclei distribution. We see that so we see that the apical basal length becomes 2.3-fold times bigger. The number of layers increases 2.5-fold. Also, the apical domain is always straight, and the mitoses are always at the apical side. So to study the influence of the nuclear movements and the increase in cell number, we consider different scenarios. So for the nuclear movements, we first consider that we consider that the cell number is constant by excluding always one cell at division. And then we consider three scenarios of no movement, low movement, and normal movement. This is achieved by controlling the time life of the cell and the duration of active G2 and mitosis. Then we study the increase in cell number by considering normal nuclear movements and considering exclusion rates of cells at division of 0%, 30, 40, or 50. So 50% leads to constant cell number over time. Then the contribution of the nuclear movements are can be assessed using the similar quantifiers that we do in the model. So apical basal length. Okay, maybe I just focus in two observations. One is that the nuclear movement control apical position of mitosis. We see it here in this plot that the divisions occur apically if in the normal nuclear movements. And apical nuclear crowding, we see that the average distance between basal and nucleus is increasing, is larger in the case of normal nuclear movements. So the black line is normal and the red line is no movement. Okay, then...
Sorry, Marina, but we are
yes.
running a bit out of time. Can I ask you to somehow wrap up the talk?
Yes.
Sorry, it's just a bit...
Yes. Okay, so the model allowed us to actually connect the difference, the cell number and the nuclei movement with the tissue quantifiers globally. And also it suggested the new experiments that were then performed again by Eric's team in to understand what is the to understand why elongation is not an emergent property of this mechanical system. And that the model suggests that there is an extra elongation force that is needed to explain the elongation of the tissue. Okay, and then the next big step is to, so with this project, we were able to reproduce the tissue and the healthy tissue and to generate new hypotheses. Then the next big step is to introduce deformation and study metastases, as Eric was saying. And the first step will be to define a basement membrane in a more realistic way. And this is a current work of Sarah and Stefan. And yeah, this is all. These are the Pierre and Sebastian, who worked in the minimization problem, Pierre and Sarah in the modeling tissue, and Eric's lab in the in vivo and in silico experiments. Thank you, and sorry for the delay.
Okay, thank you very much. Okay, so now we have time for questions. You can write on the chat, or as always, you can unmute yourself and directly ask. Okay, so maybe let me start one question for Eric, and I hope you don't hate me for this question, but you know, to write the next grant, it's something good to know. So imagine that we're in a scenario where we understand which are the mechanical factors leading to these metastases or this migration, right? Which are exactly the scenarios leading to it. Do there exist like existing therapies that could benefit from knowing this, or it will be something completely new point of view in this sense?
Well, going all the way to therapy is going to be tricky, but you could imagine it in two ways. So one way would be to detect first based on expression profile of these genes in a patient. You know, you take a biopsy from a primary tumor, and based on the expression profile, you can make some assumptions based on the heterogeneity that you can observe, because now you could imagine having access to single cell sequencing, and so you will have a map of the heterogeneity in the tumor, and knowing that you have a different collection in each patient, then you could make prediction in terms of diagnostic from patients to patients based on this knowledge. Then from therapeutics, yeah, ideally you would be going that way, trying to block this plasticity either for the cells to spread out from the primary tumor or to prevent settling in new organs. But that's really far off for the moment.
Okay, but it's good to know. Okay, thank you very much. More questions? Write on the chat or unmute yourself. Okay, maybe Osvaldo wanted to ask.
Yeah, I actually have a question out of curiosity to Eric. So this interkinetic migration that you were process that you were talking about, and this heterogeneity, which is a consequence of this, do you know how this depends along the anterior posterior axis of the neural tube in the chicken embryo?
So yeah, you have different type of heterogeneity along the axis. So if you look just at the interkinetic movement, so the fact that this nuclei would go apically to divide, you have it all along the line of the axis, but it's not linked to the epithelial plasticity in the same way. So you have different mechanisms in the more anterior part versus the more posterior part of the body. So what happens is that in the most anterior region, you have massive transformation of these cells. So you have like hundreds of cells at the same time converting into a mesenchyme. And this is not synchronized in some way with this interkinetic movement.
Okay.
So it's more random. If you look in the trunk, it's synchronized. So most of the cells that do this conversion are at a specific phase of the cycle.
I see.
So basically what people believe, what the model is that they do the extrusion toward the basal side. So they want to reach the extracellular matrix. So the fact that they are in S phase, so they are duplicating their DNA material, preparing for new mitosis, is actually an advantage because at that part of the cycle, the nucleus is basal.
Right.
And because it's the biggest part of the cell, it creates an opportunity. So you have most of the material on the basal side, and that favors extrusion toward the matrix.
Right. Okay. Okay. Thank you very much.
Thank you
Just
for
one
your
thing, just one more thing, you could imagine that with those examples of ectopic mitosis we showed, that in this case you could bypass this relationship with the S phase, because if you have a mitosis occurring in the basal, then you can imagine that the daughter cells will have more access to the matrix, being already placed in the basal side of the tissue. If they divide directly there, then they are already facing the matrix. So that's another way of seeing it.
Yeah. Very nice.